Welcome to the channel. This video is the third one of my May 2021 IB Science Multiple Choice Paper series. I have previously done chemistry time zones 1 and 2 and did reasonably well, only messed up one question. Biology is a little bit more difficult, I must confess. There's always like subjective interpretation. Some of the wordings are trickier, like stuff like evolution, like graph interpretation. I still typically do fairly well over the years, only losing 1 or 2 or 3 questions maybe, but hopefully we can do better than that here. So this is the time zone 1 multiple choice. This will represent a genuine attempt. I've not seen the paper before. I've not seen the mark scheme before. All right, so without further ado, let's jump into the paper. So in mammals, mature red blood cells are specialized in that they lack nucleus, mitochondria, or ribosomes. What statement applies to red blood cells? No chemical reactions. Well, they can still have enzymes that were previously made, but then they just won't be able to make more enzymes, I'm guessing, without mitochondria, because mitochondria releases energy that's required for a lot of other biological reactions. But there can be some passive processes, like for example, we know that carbonic anhydrase is an enzyme that's already inside the red blood cell. Having no ribosome means that we can't produce new enzymes, so I'm guessing B is the correct answer. But chemical reactions can still happen using the existing enzymes. Or there can be some other like reaction that does not require catalysis. Materials can enter or exit red blood cells. That's totally wrong, we know. White blood, well, material enters and exit depends on a lot of passive processes. But in any event, one should be the question. Answer the question one should be B. Question 2 is about red blood cells again, showing samples of red blood cells they were placed in different concentrations. What process explains the observation shown in the images? Should be mostly osmosis, because water gain and water loss have a significant impact on the shape of the cells. So solution 1, water has left the cell via osmosis because mostly you're putting the cell in surrounding salt solution, which sucks water out. And turns out the solution 1 should be the one that has the highest salt concentration, because you need excess solute in the surroundings that provides a low water potential environment, which encourages osmosis in that particular direction out of the cell. Question 4, Davson-Danieli model of the membrane structure proposed that membranes were composed of a phospholipid bilayer that lies between 2 layers of globular proteins like this. What evidence supported this model? I think this is just a matter of knowing your fact, and I think A is the correct answer already. B is actually the experiment that disproved the model in favor of the current Singer-Nicholson phospholipid bilayer model with integral proteins. C, that's definitely not correct. D is definitely not a fact. Hydrophobic region should be not in contact with water. Okay. 5, which living structure is an acceptance to cell theory? And I think you just need to know your IB. Striated muscle cells, I think, is the correct answer already because they contain multiple nucleus. Single cell alga carrying out all the functions of life. Suspicious. Okay. C and D are definitely not correct. Striated muscle fibers. But the single cell giant algae carrying all the functions of life is. Well, a single cell carrying out all functions of life is not really an exception. I think it's the giant algae having connected cellulose and multiple nucleus as well. So I think I'll still stick to A. Okay. 6, which statement applies to cholesterol? Is hydrophobic for sure. It's found on the outside of the phospholipid bilayer, which is kind of true for the most part. It's kind of embedded within the phospholipid bilayer, but in terms of size, it's usually not as big as a phospholipid molecule. I think C is better. It definitely impacts membrane fluidity. At regular temperature, it makes them less fluid. B is just wrong, and then D is transported in association with lipoproteins and not glucose. 7, which distinguishes cellulose from glycogen and starch. They are both polysaccharides. Cellulose is definitely not branched. D looks promising, but how about A and B? No starch is definitely found in plants like potatoes and rice. Only cellulose is made of glucose monomers. That's wait, no, A is glycogen. Oh, there are 3 sugars: cellulose, glycogen, and starch. Okay, so A is wrong because starch is found in plants. Cellulose is made of glucose monomers. Not true because all 3 are actually made of glucose monomers. Should be D. Okay, 8. The graph shows the progress of the same enzyme control reaction at 2 different temperatures. Total amount of substrate converted to products. Straight line, and then another straight initial section that curves and levels off. Clearly supported. The amount of product produced initially occurs at a lower rate at 40. No, not true, because 40, the graph of 40 is steeper. So more product produced over the same amount of time. So A is clearly wrong. B, you can say that 40 is kind of better than 25, but you can't say that it's the optimal temperature period. Lower the temperature, the slower the rate of reaction. That's definitely true at first. By the end, it's possible that you just use up all the reactants and then all the substrate, and that's why the reaction rate levels off. The nature at 40 degrees, I don't think that's correct because the graph has a gentle gradient, but the gradient is still positive. So I think C is the best choice. 9 was the difference between aerobic respiration and anaerobic respiration in yeast. A is definitely wrong because both processes require lots of enzymes. B is definitely wrong because both types of respiration requires some respiratory substrate. C, anaerobic, C looks promising. D, no, aerobic respiration would not produce oxygen. Answer is C. Shows atmospheric oxygen levels over time over a long history of the Earth. So about 2.5 billion years ago, a significant rise in atmospheric oxygen occurs, most likely due to the evolution or development of plants. Photosynthesis by non-vascular land plants. I want to say it's photosynthesis by vascular land plants because they have much higher biomass. I don't think this bit of vascular versus non-vascular land plant detail has been tested before. So I must confess I'm not 100% sure, but I think vascular land plants are typically the more dominant plants, and I'll just go for B, even though I don't feel too good about it. We'll find out. Okay. What genotype would be normally found in a gamete? A gamete is a hapless, it should only have 1 copy of a particular gene. So something like RR implies it's a diploid. RSTT looks like a gamete that has undergone non-disjunction. But B looks reasonable. It looks like they're talking about 2 genes that are either linked or unlinked. But only 1 allele per gene. Okay. What statement applies to meiosis and mitosis? Meiosis occurs in a greater number of locations. That's highly unlikely because only sex cells undergo meiosis. Separation of chromatids occur both during meiosis and mitosis. That seems reasonable. C, recombination occurs in meiosis, but not mitosis, so C is wrong. Reduction in the chromosome number occurs in meiosis, but not mitosis. B. 13, over time, the hull of a sunken ship may become colonized by a wide range of marine organisms. What term is used to describe all the organisms living on a sunken ship? I would say that's just the organism, so without the environment, so that should be just the community. And they say it's a wide range of marine organisms as opposed to a single species. 14, the diagram shows energy flow between 5 sinks of a terrestrial ecosystem. In a typical terrestrial ecosystem, which trophic level would have the highest biomass should always be the autotrophs. Because the higher up you go to, the higher up the trophic levels or up the food chain or up the food pyramid you go, the smaller the amount of biomass remaining, because most of the biomass would have been lost back to the surroundings as carbon dioxide through respiration. 15, what are the evolutionary origins and functions of homologous structures? Evolutionary origin should be common, but then they should end up with different function. I think the standard answer is different function, but all four choices have same, so I'm guessing B is the best. The opposite of homologous structure will be analogous structure, so different origin but same function. Okay, 16, the locust is an arthropod for invertebrate groups. What recognition feature is found only in arthropods? So jointed appendages should be the one. The exoskeleton, I guess crustaceans is also one type, but I think jointed appendages should be the answer. Because many other things have wings, and then birds have wings. Segmented body, like annelids have segments as well. 17, a dichotomous key can be used to distinguish in 4 types of plants. Which of the plants could be a bryophyte? So this is a moss, and mosses don't even have vascular tissues, because fern, conifers, and other flowering plants are the other 3 classes that you have to know for IB. The ferns, well, actually, I mean the mosses do not have vascular tissues. 18, a fluid sample is taken from the digestive tract of an mammal. The sample is basic and able to digest starch and proteins. Most likely this is from the intestine. Stomach extraction should be acidic. Mouth extract should be unable to digest proteins. Gallbladder should not contain any digestive enzymes. 19, an individual was presented with a stimulus resulting in the release of epinephrine. So like a hormone like adrenaline. So most likely nature of the stimulus. A sounds reasonable. So I mean, D sounds reasonable. A, coach shouting at the beginning of physical activity when you get hyped to do things, do sports, compete. A, I think is melatonin. Image of a close friend. Not sure what that's for. Okay, intake of glucose should stimulate the release of insulin instead, so I just choose D. 20, property of arteries. Elastic walls look reasonable already. They don't have valves. Veins do. All arteries carry oxygenated blood. That's true except for the pulmonary artery. Arteries receive blood from the atria. It's not true for the pulmonary. The arteries receive blood from the atria. No, ventricles. Okay, of course. Okay. 21. Okay, what process results in the exchange of gases across membrane of pneumocytes? It should just be simple diffusion, and that's just a matter of knowing your facts. What statement applies to an axon at rest? That should be a. The A is definitely wrong because the resting potential is -70. Outside more positive than inside. The external surface is positive relative to the internal surface, so B looks reasonable. And then internal surface is got higher concentration of sodium ions. That's false because the sodium potassium pump actively pumps sodium outside. So you want sodium plus waiting to come in. Like once the voltage gated sodium channels open, but then the resting potential is features outside being more positive than the inside for sure. 23, what structural feature enables saltatory conduction? And that's referring to the nodes of Renvia between myelinated sections. Should just be A. And the Schwann cells are the ones that have the myelin. But then, so with these, within these myelinated axon sections, the action potential is super fast. At the nodes of Renvia, that's where all the like membrane proteins act in order to maintain a sufficiently high sodium ion concentration. Okay, 24 is the menstrual cycle, which line on the graph represents progesterone. So it should be D because progesterone is a hormone that peaks near the end of the entire cycle. What event occurs approximately at the time of the dotted line? That's not really dotted, but whatever. So time X should be ovulation because that's shortly after hormone A, which is LH, peaks. So the answer is just B. 26, what regions of DNA call for the production of a specific protein? Just for genes, but of course they try to make things tricky, but then it should be the exons because exons are the non-junk sections because the introns are the ones that are cut out. Exons are pieced back together to correspond to the mature mRNA, which is the genetic material for, well, I guess, which region of DNA. Now, I guess DNA can still have you can still have exon region on the DNA, I guess. The other ones I have some reservations. I'll still go for exons, even though typically we only refer to exons on like the mRNA transcript. Okay, 27, which statement applies to tRNA? At least one type of tRNA. I think A sounds reasonable. B is wrong. Again, like tRNA is like shape specific. There needs to be different tRNA for different amino acids, and there are 20 of them at least. tRNA carries its main role in the cytoplasm because that's where the translation mechanisms are. tRNA is not produced. tRNA assists translation, so A should be the correct answer. 28, which statement applies to the tertiary structure of the enzymes? Choice A is wrong because the correct structure would be the primary structure. B should be wrong because all enzymes need to have some 3D structure in order to bind to the active sites. In order to correspond to the active site which the substrates can bind to. An example of a tertiary structure in an enzyme is the alpha helix. That's wrong because an alpha helix is a secondary structure. The answer should be D, which is a correct statement itself. Okay. 29, decarboxylation. So the loss of CO2. So clearly choice A is correct. B is more like carboxylation because you're combining CO2 with stuff. C, you release CO2, but that's just the overall combustion process of methane. D is condensation, so not correct. Okay. 30. Succinate dehydrogenase is an enzyme that catalyzes the oxidation of succinate acid. If malonic acid is added to the mixture, a reduction of rate of reaction corresponds to malonic acid being some kind of inhibitor. And if you add succinate acid, so the succinate acid is a desired substrate. And if you increase the concentration of succinate, that means that, and then resulting in the restoration of the rate of reaction, that means that malonic acid is like a competitive inhibitor, which dissociates reversibly. So the answer is just D. 31, where does electron tomography allow mitochondria researchers to do? Electron tomography just locates like electrons, which is not saying much, but then I think C should be correct because you can locate the molecules that are temporarily carrying these extra electrons. Images of crystal, you just use micrography instead of tomography. Because tomography is like locating like regions which are rich in electrons and regions which are poor in electrons. Molecules, ATP synthetic molecules, you need X-ray crystallography because those are proteins. I don't think you can actually ever visualize oxidation reduction reactions. 32, which process does not take place in the stroma of chloroplasts? Stroma of the chloroplast is where light independent reaction takes place. So fixation of carbon is correct, and synthesis of carbohydrate should be correct. Reduction of NADP occurs at the surface of the thylakoid membrane. But the synthesis of the RUBP, which is a chloroplast enzyme, because the chloroplast definitely has its own ribosomes and circular DNAs. I'm guessing I'll just go for C. Could you reduce NADP during the light-dependent reaction at the end of the electron transport chain? I'm guessing that occurs on the thylakoid membrane. So we'll see. Question 33, a summary diagram of photosynthesis is shown. Which molecule represents ATP? Well, ATP is generated early on during a light-dependent reaction. And then it's used as an energy source for carbon fixation. So I think A is the most reasonable answer. D could be some of the C3s and C5s that are intermediate. But actually, A is most likely, what's A? I mean, what's D? Well, C would be oxygen. I guess D can be ADP. And then B would be carbon dioxide. 34, which method can be used to induce short-day plants to flower out of season? We need to make sure that they have lots of darkness. And you want to grow them, cover them with opaque cloth, just give them more darkness. And darkness, well, light would most likely be an inhibitor of like growing floral pots. So I think D should be correct. Winter and summer, I think these are too vague. It's like several hours, that's a significant difference. In terms of the significant reduction of daylight that they typically receive. We'll just hope that D is correct. Okay. 35, valid statement regarding chromatids. Sister chromatids separate during meiosis. 1, that's not true. Homologous chromosomes are the structures that separate. Here's another form between non-sister chromatids, and I think that's correct. Crossover is exchange of DNA between non-sister chromatids, so C is wrong. Non-sister chromatids don't necessarily have the same combination of alleles, and specifically, if you're heterozygous for a trait, then for sure the non-sister chromatids have different alleles. You get a different allele from each of your parents. Okay, 36. What do we have? So most likely we have a linked genes situation. Homozygous vestigial winged black-bodied fly. So I think those that will be double recessive. So double recessive crossing double heterozygous should be a situation where you get 1, 1, 1, 1, like 25% across, but here you don't get 1, 1, 1, 1. Because for the I'll just draw all the grid maybe. Because for the homozygous recessive individual, it would be small b without a plus, and the VG without a plus. Whereas the double heterozygous parent would have the ability to produce 4 different types of gametes with respect to these 2 genes. So the first one is double dominant. Second one is dominant in color and recessive in wing size. Actually, I'll just draw the complete genotype for them. Anyway, that should be a 1 to 1 to 1 to 1 distribution, which is clearly not observed. And the most likely reason for deviation is that the 2 traits are on the same chromosome. So in other words, they are linked genes. The traits on, so which statement is correct? The predicted phenotype ratio should be a 1, 1, 1, 1 situation. There is independent assortment of wings, but not body color, don't think so. I think C should be correct because they have 2,300 as a number given, and if you divide it by 4 or multiply it by 25%, same thing, you should get 575. 37, that looks like a sarcomere. Which structure is myosin? Myosin would be the thick filament. So B looks like the myosin head. I was expecting them to label the middle horizontal bars lines because A will be the actin and C will be the tropomyosin and D is the Z line. So I'll just go for B. Okay, yeah, 38 was normally found in the urine of a healthy individual. Definitely not glucose, definitely not red blood cells. Sodium ions, I would say. Proteins, no, proteins should not be in urine because proteins are too big to go through the urine or the kidney filtration mechanism, like the glomerulus. Okay, 39, pregnancy test for humans based on detection of hormone HCG, which is produced by the embryo. So C should be the correct answer. D, expansin is a plant protein that loses connection between cellulose fibers and plant cell walls, allowing growth. In what location would the expression of expansin gene be expected to be increased? So it's basically the details of auxin action. I don't think expansin itself is in the syllabus, but I'm guessing you have to recognize that expansin synthesis is promoted by the presence of auxin, because auxin allows cell walls to be loosened. And that should be on the shade side of a shoe being exposed to light, because light causes auxin to migrate to the opposite side. All right, that's it. So moment of truth. Let's see which questions I messed up, if at all. Okay, let's see. BDBA, all good. CDCC, I messed this one up for some reason. This is so sad. But I wanted to change the color to font color. BBAAB, the rest is fine. BACDA, this is fine. BBADB, all good. CADAD, all good. CCADB, I messed this one up. That's very annoying. CBDCA, so the rest is fine. So I got 38 out of 40. It should still be a comfortable session, but anytime you get things wrong, it's kind of annoying. So let's review the questions. So question 10, I think, was the photosynthesis. So what causes photosynthesis? Oxygen produced by the photosynthesis. I didn't even consider that option because I thought those were present originally in the ocean, but then the amount is not high enough. Because I guess the collective biomass is still quite significant. They clearly, like, the evolutionary path should be synthetic photosynthetic bacteria before you get plants that actually have chloroplasts because of endosymbiotic theory. All right, so that's my fault for not knowing my stuff well enough. But that's how you learn. Okay. 31 I actually got wrong. Movement of electrons in the electron transport chain. I guess these are too small of a structure, similar to, yeah, for sure, electron transport chains are embedded within crystal. So I'm guessing you can see where the electrons are, but then you can't see how they move too much. All right, so that's it. So that's 2 questions wrong, but I hope you guys learn from the rest of the video. So I'll next shift my focus to the other paper 2. So the other time zone 2 in my next video. So thank you for watching.